Hello student, this is Shumona from Simmi Foundation. Today we are gathered to learn about a new topic named antigen. So first of all we have to know that what is antigens. Adaptive immune responses arise as a result of exposure to foreign compound. The compound of foreign origin that evokes the response is referred to as antigen, a term initially coined due to the ability of these compounds to cause antibody responses to be generated. An antigen is any agent capable of binding specifically to a TCR or antibody molecules. Any substance which and when introduced parenterally and into the body stimulates the producing of antibody specifically. This is a traditional definition since it had got some exception. The exception is polio vaccine, which is orally administration. Some antigen may not produce antibodies. Specificity is the hallmark of all immunological reasons. To understand the topic, we have to know about the structure of antigen. Antigen, the smallest unit of antigenicity, antigenic determinant is epitope. Consists of 4 to 5 amino residues or monosaccharide residues. Possesses a specific chemical structure, electric charge, and spatial configuration capable of sensitizing and immunocytes of sensitizing. Here is a term coined paratope, area of antibody molecules on which epitope binds. This produces specificity of antigenic reaction. Now we have to know the biological classes of antigen. Depending on the ability to produce antibody formation, antigen are classified as T cell dependent, which is shortly TD, structurally more complex, and T cell independent, which is shortly TI, simple limited number of repeating epitopes. Although antibody is produced by B lymphocyte, but it requires cooperation of T lymphocytes. Now we have to know about the properties of antigen. First of all, the size of antigen is related to the molecular size. Larger molecules are highly antigenic, whereas heptanes are low molecular weight having low antigenicity. The chemical nature of antigen is directly proportional to degree of structural diversity. Amino acid, monosaccharide, lipid nucleic acid, which is the chemical structure of antigen. Susceptibility of antigen, only substance metabolized by tissue enzyme to epitope fragments potential to produce antigen. Polystyrene is not antigenetic. Also, antigen rapidly broken down doesn't show antigenicity properties. D amino acid are not metabolized in the body, whereas L amino acid do prevention of autoimmunity. Here is a term which is origins described by Ehrlich. Normal body contain numerous antigen or antigen producing substances which may not produce antibody, but introduction of these into other body produce immune responses. This response is directly related to degree of foreignness. Antigen from some specific show less antigenicity than others. Antigen shows various types of specificity. They are mainly antigenic specificity, species specificity, isospecificity, auto, organ, and heterogenic specificity. What is antigenic specificity? Chemical nature of antigen makes it different from other. It may arise on the difference of group attached on ortho, meta, or para position. also on the basis of spatial arrangement, dextro, levo, and meso. Specific specificity, all individual contain specific specificity antigen. This has been used in tracing evolutionary relationship. Example, a individual sensitive to hot serum will be sensitive to the equine but not with bivalve. What is isospecificity? Antigens found in the some but not all members of species. Blood groups systems is based on this genetically determined. And what is also autospecificity? Sometimes autogenous antigens starts showing antigenicity. Autoimmunity disorder like rheumatoid arthritis, Sjogren's syndrome. And now we have to know about the function of antigen. Immunogenicity, induction of immune response, immunological reactivity, which is specific reaction with antibodies or sensitized cells based on the function of antigen is classified as complete antigen, induced antibody formation and specific immune response, partial antigen, which is hapten, a specific non-protein substance incapable of inducing antibody formation itself but elect immune response when coupled with. So in this video we know about the structure function of antigen. This topic is slightly difficult to understand, but if you hear the topic details and see the slide details, you can understand it easily. So student, I think you can understand this topic. For today, that's all. Thank you and keep learning.